Hello everyone. Today let us learn about the pollination mechanism, which is a common phenomenon for the reproduction in angiospermic plants. See, now what is pollination? How one can define a pollination? It is nothing but the transfer of pollen grains from anther to the stigma of a flower. Pollen grains are immobile, as we all know very well. They cannot reach the stigma by themselves. Means they cannot move on their own. The pollen grains, which are mentioning, they cannot move on their own. An external agent is required for this pollination mechanism. It can be like wind, insects, etc. Now let us differentiate the pollination mechanism in different plants based on what we are differentiating and what are the types of pollination can be seen here clearly. See, here the pollination is of two types, that is self-pollination and cross-pollination. In self-pollination, again we can see two different types of mechanisms. The first one is autogamy and the second one is genetogamy. Autogamy is the phenomenon which takes place in the same flowers. Means the two organs of a flower is included in one individual flower. Whereas the genetogamy, here we can see the different flowers of same plant. It means involvement of two flowers can be seen in this type of pollination. Now, in cross-pollination, we can see two types of agencies again. Cross-pollination can also be known as xenogamy, and these abiotic agencies are the first type of pollinating agents. The first one is anemophily, that is, the transfer takes place through wind. Second one is hydrophily, the transfer takes place through water. Whereas the biotic agencies, here the biotic agencies can be of like insects, birds, snails, etc. So the first one is entomophily, second one is pyrotrophily, third one is aeromophily, last one is ornithophily, that takes place by birds. Now let us learn what is self-pollination in detail. It is nothing but the transfer of pollen grains from the anther of a flower to the stigma of either the same or genetically similar flower. Means the flowers of the two individual plants must have the same genetic appearance, or the flowers which are belonging to the same plant should show the same genetic characters. Accordingly, self-pollination is of two types, autogamy and genetogamy, as we have shown in the picture. What is autogamy then? It is a type of self-pollination usually in which an intersexual or perfect flower is pollinated by its own pollen. Means the pollen is from the same flower. Autogamy occurs by two methods. Generally, they are homogamy and cleistogamy. Homogamy means the anthers and stigmas of chasmogamous or open flowers are brought together by growth, bending or folding. Right? The twigs of a flower can bend sometimes. The twigs of flowers can. Whereas in cleistogamy, cleistogamous flowers, the anthers devise inside closed flowers. That means all the organs are in closed condition in the cleistogamous flowers. Growth of style brings the pollen grains in contact with stigma. Pollination and seed set as assured. Pollinations are not required. Means cleistogamous flowers can obey in terms of the pollination times. Let us see the self-pollination mechanism, how the pollen grains can be seen in open flower, the pistil of the same flower, how it is collecting the pollen, and how the pollen comes loose from the stamens. Now, what is genetogamy? It is a type of pollination again in which pollen grains of one flower are transferred to the stigma of another flower, belonging to either the same plant or genetically similar plant. Again, we can see the two different type of plants. In genetogamy, the flowers often show modifications similar to ones found in xenogamy or cross-pollination. These are two different flowers which are present on the same plant roots. Now let us learn what are the advantages and disadvantages of the self-pollination. The self-pollination, it is advantageous in maintaining the parental characters like purity of the race indefinitely, and it is also advantageous in self-pollination, and it is used to maintain the lines for hybridization experiments. The third advantage is plant does not need to produce large number of pollen grains, and the flowers do not develop devices for attracting insect pollinators. Now, what are the disadvantages in this self-pollination mechanism? Useful characters are seldom introduced. Vigor and vitality of the race decreases with prolonged self-pollination. If we do the self-pollination for many generations, we can lose the vigor and we can also lose the vitality. Now, immunity of diseases can decreases. Adaptability to changed environment are reduced. It is a bad thing which can be done when we try to use the self-pollination all the time. Now let us learn the co-evolution of flower and its pollinator species. Means how the pollinating agent and the flower can evolve for a longer generations. Co-evolution is the evolution of two species that interact extensively with one another so that acts as a major source of natural selection on the other. When other evolves a new feature or modifies itself, the other evolves new adaptations in response of it. Means, if the flower is able to modify its features, it is also able to adapt in another twice. This constant mutual feedback modification between the two species is known as co-evolution, because both the things are evolving at the same time simultaneously. The co-evolution of a flower of the pollinator species can be tightly linked with one another, because if the plant is not adapted, it cannot show any new modification. If the plant is in adaptation of new practice, it can show the new thing in a vice phenomenon. Let us learn a unique species of orchid in showing the co-evolution. Dysanindia is a rare orchid found only in a few places of South Africa. And until Johnson came to study, he is the person who studied it for the first time in Dysanindia, that is co-evolution. No one knew how it is now pollinated. After a lot of patent orchid watching, he and his colleagues discovered that it is visited exclusively by the fly shown in the picture. Here we can see the Dysanindia. Its proboscis is well matched to length of orchid. See, here we can see the proboscis of this insect, which is a pollinating agent. It matches the length of the orchid. And the orchid throws pollen in just the right place so that they get stuck to the fly. When the fly is fixed on this flower, it gets stuck with the length of the orchid. In this picture, the two diamond yellow packets of the flies now, this fly is named as orchid fly. So, here we can see the yellow snout and this yellow color of the orchid flower. So, this fly is named as the orchid fly since it is showing yellow packets on the fly snout. This is the snout part. Thank you.